Let's learn about the advantages of chromatographic technique. Chromatography is a highly versatile technique that can be adapted to a wide range of applications. It is highly sensitive and can detect trace amounts of analytes. Chromatography can provide highly accurate and precise results. It can be used to separate complex mixtures of compounds with high resolution. Chromatography is a relatively fast and cost-effective technique for separating and purifying different molecules. Disadvantages of chromatographic technique. Chromatography can be time consuming, especially if multiple steps are required for separation and purification. It can be difficult to select the appropriate stationary and mobile phases for a given sample, which can affect the accuracy and precision of the results. Chromatography can be expensive. And now we are going to learn about the principal parts, working and applications, and advantages and disadvantages of column chromatography. Column chromatography is a widely used technique in biochemistry for the separation and purification of various biomolecules. It is based on the principle of differential partitioning of solutes. Differential partitioning of solutes between a stationary phase and a mobile phase. In this technique, the stationary phase is typically a solid matrix or a liquid bound to a solid support, while the mobile phase is a liquid or a gas. The principle of column chromatography is based on the separation of components of a mixture based on their differential partitioning between stationary phase and the mobile phase. The stationary phase is usually a porous solid matrix packed into a column. The stationary phase is usually a porous solid matrix packed into a column, and the mobile phase is a column liquid that passes through the column. As the mobile phase flows through the column, the components of the mixture partition between the mobile phase and the stationary phase. Components that have a higher affinity for the stationary phase will spend more time in the column and will elute later, while components that have a higher affinity for the mobile phase will elute earlier. Now let's learn about parts of column chromatography. The column is the main part. The column is the main part of the chromatographic setup. This is usually made of glass or plastic and can be of different lengths and diameters depending on the sample size and the separation required. Stationary phase is there. The stationary phase is a solid matrix or a liquid that is immobilized on a solid support, and the stationary phase is chosen because and based on its ability to selectively retain or separate the desired components from the mixture. Then there will be a mobile phase. The mobile phase is a liquid or gas that flows through the column and carries the sample components with it. The mobile phase is chosen based on the polarity and solubility of the components to be separated. Sample injection port will be there. This is the point where the sample is injected into the column. Then detector will be there. The detector is used to monitor the elution of the components from the column. Sample injection point is a very important one, and a detector will be there and working. In column chromatography, the sample is loaded into the top of the sample injection port of the column, and the mobile phase is passed through the column. The sample is loaded onto the top of the column and the mobile phase is passed through the column. The sample components move down the column at different rates depending on their affinity for the stationary phase. The elution profile of the sample is monitored using a detector, and fractions of the elute are collected at different time intervals. The fractions are then analyzed to determine the purity of the individual components. Now, column chromatography is used in a variety of applications, including separation and purification of proteins, nucleic acids, and other biomolecules, purification of natural products and synthetic products, analysis of complex mixtures such as crude extracts from biological samples, etc. are the advantage applications. Next, advantages. Column chromatography has several advantages over other separation techniques such as high resolution and separation efficiency. High sample capacity and throughput, versatility and compatibility with wide range of solvents and stationary phases. Disadvantages are column chromatography also has some disadvantages, including limited scalability, potential for simple degradation due to prolonged exposure to the stationary phase, Sample degradation will be happening due to the prolonged exposure to the stationary phase. High cost of equipment and consumables. Now let's learn about the principal parts, working and applications of advantages and disadvantages of paper chromatography. Paper chromatography is a type of chromatography that uses a piece of paper as the stationary phase to separate and analyze mixtures of different compounds. The principle of paper chromatography is based on the differential partitioning of the components of a mixture between a stationary phase and a mobile phase. The paper contains microscopic pores that act as a stationary phase, and the solvent travels to the pores and separates the different components of the mixture based on their solubility and partition coefficient. Now let's learn about the parts of paper chromatography. Paper strip. Paper strip. A piece of paper strip acts as a stationary phase for separation. Then the sporting area will be there. The sample is sported on this area. Solvent front. The point where the solvent reaches after moving through the paper strip. Next, solvent, the mobile phase that moves through the paper strip. Container, the apparatus that holds the paper strip and the solvent. Working of the paper chromatography. The paper strip is sported with a sample to be separated and placed on a container containing the mobile phase. The mobile phase moves through the paper strip and carries the movement components of the mixture with it. The speed of each component depends on its solubility and partition coefficient. Components that are more soluble in the mobile phase will travel faster than the less soluble components, resulting in separation of the components based on their relative solubility. Now, applications of paper chromatography: separation of amino acids and sugars, separation of plant pigments, separation of food dyes, analysis of drug purity, separation of inorganic ions, analysis of environmental pollutants, etc. are the applications. Now, what are the advantages of paper chromatography? Simple and inexpensive technique, can be used for separation of a wide range of compounds, requires a small amount of sample for analysis, does not require any special equipment or expertise, can be used for both qualitative and quantitative analysis. These are the advantages. Now let's learn what are the disadvantages of paper chromatography. Low resolution and sensitivity compared to other chromatographic techniques. Time consuming process, limited separation capacity, poor reproducibility, limited sample loading capacity. These are the disadvantages.